This is glycolysis pathway, and this pathway is divided into three distinctive phases. First one is energy investment phase, second is splitting phase, and third one is energy generation phase. First is energy investment phase. That is for the first step, that is phosphorylation of glucose. Here, the glucose is phosphorylated to glucose 6-phosphate in the presence of enzyme hexokinase or glucokinase, and it utilizes one molecule of ATP and in the presence of magnesium ion. In this process, it consumes one ATP molecule, that is energy investment phase. That is, it consumes one ATP molecule. The second step is isomerization. Isomerization of glucose 6-phosphate to fructose 6-phosphate. Here, the glucose 6-phosphate is converted to fructose 6-phosphate in the presence of phosphohexoisomerase. Phosphohexoisomerase enzyme. Next is phosphorylation of fructose 6-phosphate to fructose 1,6-biphosphate. Fructose 1,6-bisphosphate. In the presence of enzyme phosphofructokinase. In the presence of enzyme phosphofructokinase, and this step also utilizes one molecule of ATP in the presence of magnesium ion. The process is also take place in the presence of magnesium ion and phosphofructokinase, and it utilizes one molecule of ATP. So in the first stage, that is energy investment phase, it utilizes 2 molecules of ATP. Next is splitting phase. Here, fructose 1,6-bisphosphate is break down to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it is called as splitting phase. Here, a 6-carbon compound, that is fructose 1,6-bisphosphate, is split into 2 3-carbon compound, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. The enzyme phosphotriose isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That is phosphotriose isomerase, which interconvert dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Thus, 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecule of glucose. 2 molecule of glyceraldehyde 3-phosphate is obtained from 1 molecule of glucose by the interconversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, and also 1 molecule of glyceraldehyde 3-phosphate is obtained from fructose 1,6-bisphosphate by the enzyme aldolase. And this is the splitting phase. Next is energy generation phase. Here, the glyceraldehyde 3-phosphate, that is converted to 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate or 1,3-diphosphoglycerate. Here, the glyceraldehyde 3-phosphate is oxidized to form 1,3-diphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase and form 1,3-bisphosphoglycerate. And this enzyme requires the presence of NAD+. The presence of NAD+, and the NAD is converted to form NADH plus H plus. 1 NADH is equal to 3 ATP. 1 NADH is equal to 3 ATP or 2.5 ATP. So this process is carried out in the presence of NAD and glutathione. NAD and glutathione. In aerobic condition, this NADH passes through electron transport chain and 6 ATP are synthesized by oxidative phosphorylation. So in this step, ATP is generated. Next is transformation step, that is 1,3-diphosphoglycerate or bisphosphoglycerate is converted to form 3-phosphoglycerate. 3-phosphoglycerate in the presence of enzyme phosphoglycerate kinase. And the process is happened in the presence of magnesium ion and it utilizes ADP and form ATP from ADP molecule. So in this step also 1 ATP molecule is generated. Next step is isomerization. Here 3-phosphoglycerate is changed to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. Phosphoglycerate mutase. And in next step, the 2-phosphoglycerate is converted to 2-phosphoenol pyruvate. 2-phosphoenol pyruvate in the presence of enzyme enolase. In the presence of enzyme enolase and also in the presence of magnesium ion. It also require ADP molecule and generate ATP. It generate ATP. So the eighth step is conversion of 2-phosphoglycerate to 2-phosphoenol pyruvate in the presence of enzyme enolase and in the presence of magnesium ion 2-phosphoenol pyruvate is formed. Next step is conversion of phosphoenol pyruvate to 2-pyruvate. The phosphate group in the phosphoenol pyruvate is transferred into ADP molecule in the presence of enzyme pyruvate kinase, in the presence of enzyme pyruvate kinase and magnesium ion with the formation of ATP and pyruvate. Here the phosphoenol pyruvate is converted to pyruvate in the presence of enzyme pyruvate kinase and it utilizes ADP molecule and it generates 1 ATP molecule in the presence of magnesium ion. Here the formed pyruvate is in enol form. The formed pyruvate is in enol form and this step is an example of substrate level phosphorylation and this reaction is irreversible. This reaction is irreversible and in this step 1 ATP is produced. Next step is conversion of enol form of pyruvate to keto form under aerobic condition. Under aerobic condition. The enol form of pyruvic acid, it is converted to keto form of pyruvic acid under the influence of enzyme pyruvate kinase. Under the influence of enzyme pyruvate kinase. That is enol form of pyruvate just converted to keto form. Last is conversion of pyruvic acid to lactic acid. Under anaerobic condition, pyruvate is converted to lactate. Pyruvic acid form the end product of glycolysis in tissues having sufficient oxygen. In the absence of oxygen, the tissues will form lactic acid from pyruvic acid. In such